Hi, everyone. How are you doing today? Thank you all so much for joining today's webinar. Today, we're going to be learning a little bit about how to track a gene in ClinGen, and we are going to have Danielle Azzariti speak with us. She's a project manager of ClinGen, so really excited for today's webinar, and I'm really excited to learn a lot more about the capabilities of ClinGen along with you all. I'm going to give it, the obligatory few minutes for everybody to join. As a reminder, our webinars are recorded and then shared on our YouTube playlist that is in a webinar format. And I will go through all of those little housekeeping tips and tricks in a minute. So this is a schedule of the webinars that we have previously had. As I just mentioned, they are all on our YouTube playlist. There is a QR code for you to be able to access them, and we will put the link to our YouTube playlist in the chat shortly. So we have done this year the importance of individuals sharing health and genomic data. Alexis Morgan led a great talk on how to read a genetic test report. We had guest speakers Maya Chopra and Sonal come and talk to us about the Brain Gene Registry, which is a patient advocacy organization that GenomeConnect partners with. And then last month, Melissa Landrum led a talk on how to search ClinVar and how to track different variants in ClinVar. And then today we are expanding on that, and we are going to talk a little bit more of how to track a gene in ClinGen. So I will just do some housekeeping items. So this is a webinar format. And if you guys have any questions, please put them in the Q&A box. It's the little double quote box image, usually towards the bottom of the Zoom screen. We will have a session at the end of the talk for Q&A, but we also try and address some of the questions that come up. So feel free to put them in during the talk, and we will get to them maybe during the talk or even after during that Q&A session. So my name is Caitlin. I forgot to introduce myself. I'm a member of the GenomeConnect team. And today we are going to talk a little bit about candidate genes and then how does ClinGen work with candidate genes and then how to track a gene in ClinGen. So for the first part of the talk, I'm going to go over what is a candidate gene, or sometimes they're called genes of uncertain significance. And then I'm going to pass it over to my colleague to go over how does ClinGen evaluate gene-disease relationships and how to track a gene in ClinGen. So Danielle Azzariti is our guest speaker today. She is a genetic counselor by training, and she is a project manager of ClinGen at the Broad Institute. And she also does amazing work with GenomeConnect. And so we're really excited for her to be here today and share her expertise. So to start off, we're just going to go over a little bit of a background. So what are candidate genes? Sometimes they're referred to as genes of uncertain significance as well. So I'm going to refer to them as candidate genes for the remainder of the talk, but I just won't let everyone know that they're sometimes referred to as genes of uncertain significance if they're called that in your report or if you hear any of the providers, researchers talking about them. So each of us has more than 1 trillion cells that make up our bodies. We have different types of cells. We have skin cells and brain cells and heart cells, and they are the building blocks of our body. In each of our cells, all of our genetic material or our DNA can be found. And our DNA is like our body's instructional manual. Each instruction is called a gene. And to keep our DNA organized in our cells, they are wrapped tightly into structures called chromosomes. And then each individual instruction, as I mentioned, is called a gene. So this is just a basic image to show how our DNA is broken down in our cells. I did this background because we're going to be focused on genetic variants today. And so we want to kind of know what is a genetic variant. Oh, sorry. So genetic variants are changes in our DNA. And while most of our DNA sequence is the same from person to person, we all have variants. Some variants can impact how those proteins work. So genes code for proteins, and proteins do everything in our body. So some variants can affect how those proteins work in the body and can cause developmental or health differences. And some genetic changes or variants are just part of who we are. One type of genetic variant is called a sequence variant. A sequence variant is a change that's really identified in one gene. If we think about our genetic code kind of like a person's own very own library, a person typical has a certain number of books in their library. And then if there is a spelling change in the sentence in one of those books, that would be considered a sequence variant. So just one small spelling typo in all of the number of books. Sequence variants are typically named based on the change to the DNA and the change to the protein. So if you're looking at a genetic report and you see a C and then a period, we call it a C dot on your report. This tells you that the change to the DNA. The C is followed by a number, so you know exactly what letter is changed in the DNA. So in the sample on the screen, there is a change at position 622. So the letter 622 in the gene CHG8 is changed from a G to an A, and that's that greater than symbol that we see. We can also call this based on the protein level change. So if you see a P period or a P dot on your report, this tells you what the change in the protein is. So in this example, that it's P.glycine 208 serine. This tells us that amino acid 208 in the protein is typically a glycine. But because of the specific change in the DNA that we saw, that C dot, this amino acid has been changed from glycine, the GLY, to serine, the SER. Another type of genetic variant is called a copy number variant. And a copy number variant means there is a large extra or missing amount of genetic material. So this can include extra or missing genetic material that can include more than one gene. So we kind of think about it going back to our library example. This would be if you were looking at somebody's library, they have a number of books that are missing. So not just a change in one sentence, a whole slew of books are missing. Copy number variants are typically named for their location. So same as sequence number variants, we like to describe where the change is. So our chromosomes usually have two arms. There's a shorter arm of which we call the P arm and a longer arm of which we call the Q arm. So using the image on the right, this 22q11.2 deletion might be used to describe a deletion on the long arm of chromosome 22. So 22 tells us the chromosome number. The Q in this situation tells us that the material is on the long arm of that chromosome, and the 11.2 tells us the band on the chromosome that we're looking at, and then deletion tells us it's a deletion. If it was a duplication, they would have said the word duplication. So all this is just to kind of go over what's the types of variants that can be identified. And then once a genetic testing laboratory identifies this variant through testing, the next step is to interpret that variant or try to determine if this change, this variant that they found, impacts how that genes work on the body. And if that change that they identified could be associated with a health condition or developmental differences. So really the first question that we need to ask when we find a change is that gene associated with disease? We found a change, it impacts this gene or multiple genes. Do we know what those genes do in the body? That's the first question. If we do, Then we can start evaluating the specific changes that we see and to determine how that change would impact the functioning of the body. If we identified a change in a gene and we don't know what that gene does, that gene has not been associated with disease, then that variant is going to be uncertain. We don't know if this change that we have identified, if this could be related to our patient's health, or if this could be a risk for any health concerns in the future. If we are uncertain about the gene's role, it is going to be considered that candidate gene or a gene of uncertain significance. We really just don't know how to interpret anything about this gene yet because we don't really know its role in the body. By sharing data with GenomeConnect, as you know, is our patient registry, and we share data with public registries, so public databases. And so if we learn about any changes to variants, meaning that we have saw that there was a different interpretation on a variant or a change in the classification of the variant, we like to share that information back with our participants. So participants who have enrolled in GenomeConnect, who have uploaded a genetic test report and have opted to receive these updates, if GenomeConnect learns about an update, we will alert the participant via email that there may be updated information regarding the results. All right, that was a quick background on candidate genes, and now I'm going to pass it over to my colleague Danielle, and she's going to tell us a bit more about how ClinGen evaluates gene-disease relationships.
Excellent. Thank you so much, Caitlin, and thank you for having me today. Thanks to everyone who's joined. So in Caitlin's introduction, she mentioned a first step of interpreting if a genetic variant is contributing to a disease is to determine if the gene the variant was found in is associated with that disease. So as Caitlin said, I'll provide you an overview of how ClinGen specifically evaluates these gene-disease relationships and then how to track that information through our website. Next slide.
Sorry, wrong way.
You're doing excellent. So first, I'll give you a quick overview of what ClinGen is, in case that's one of the first times you've heard that term. So ClinGen, which is short for the Clinical Genome Resource, is an NIH or National Institutes of Health funded project. ClinGen is the overarching effort that the GenomeConnect patient registry falls under. And ClinGen's central mission is to support a resource that helps understand what genes and variants are associated with disease and how this genetic information can inform clinical care. And we do this in the steps illustrated here. We do this by first encouraging the sharing of genetic and health data by key groups, patients, clinicians, laboratories, researchers, and this includes the sharing through our patient registry, GenomeConnect. In the second main step, we use this data to answer a number of curation questions. And once we answer these questions, we make this information publicly available, building a genomic knowledge base with the goal of improving patient care through genomic medicine. Next slide. And one of the key curation questions that you may have noticed that's pertinent to today's webinar is gene disease validity. Can variants in a particular gene cause a particular disease? So that's what we'll do a deep dive into now. Next slide. Here you go. So to answer this question, is there evidence that variants in this gene can cause this disease? ClinGen actually published a framework to systematically evaluate this. That was published in 2017, and since then, we've been using that framework and building upon it to do this work. This is a standardized way to evaluate evidence for or against a gene-disease relationship. And to do this, a curator gathers evidence from published studies about the gene, including genetic evidence from individuals who have been identified to have the variant in that particular gene, and any health effects they may have, as well as experimental information about the gene's function. All of that information is evaluated and scored according to the framework. Next slide. Based on the available evidence, a classification is then assigned to describe the strength of evidence for the gene-disease relationship. And the classifications are displayed here on the right-hand side. If a gene's role in disease has been demonstrated in the research and clinical settings and upheld over time, that gene will likely reach definitive or strong. And from there, lesser evidence may be available supporting the gene-disease relationship. And in that case, a gene-disease relationship may only reach moderate, limited, or no known disease relationship. If there's conflicting evidence related to that gene's impact on disease, it would likely receive a classification that you see at the bottom of disputed or refuted, meaning that there's evidence that suggests the particular gene is not associated with a particular disease. So what do we do with these classifications, or what do we hope the medical and genomics community at large does with these classifications? Well, they help guide genetic testing and variant classification. Variants without well-understood gene-disease relationships, such as those classified as limited, disputed, or refuted, should not be classified higher than a variant of unknown significance, given the limitations of what we know about that gene. A variance in genes with a moderate gene-disease relationship should not be classified higher than likely pathogenic per professional standards for the same reasons about the limitations of the evidence for that gene-disease relationship. Laboratories may use this type of gene curation data, gene disease validity relationship data, to inform what genes they put on a genetic test panel or what genes they evaluate on an exome or genome sequencing test. And clinicians may use this type of information when interpreting test results for their patients. Next slide. Thank you. So within our ClinGen project, groups we refer to as gene curation expert panels, or GCEPs, are busy using this classification process to evaluate gene disease relationships in specific clinical domains. For example, we have a hereditary cancer GCEP that evaluates genes that have been considered to contribute to cancer predisposition. The GCEPs include clinicians, researchers, and laboratorians who specialize in that clinical area. And once a GCEP has formally approved a gene disease curation, it is made available on ClinGen's website. To date, over 50 of these gene curation expert panels have evaluated over 2,800 gene disease relationships, which sounds like a lot, and it is, but I'll put the caveat now that we still have a lot of work to do. There's a lot of genes that we haven't curated yet, but we have our sights on doing them. Something important to keep in mind, especially for this topic that we're focused on today, gene disease classifications may change over time. Just because something is classified as moderate initially does not mean it will always stay there. As more data is collected and evaluated over time for or against that gene disease relationship, it may push that classification up or down on this schema here. Next slide. So that was a brief overview of how ClinGen is curating this information. And I will say that many laboratories who are doing genetic testing may use a process similar to this. Not everyone is using the exact same framework, but I oriented you to ClinGen's because that is the basis of all the gene curation, gene disease validity evidence results that you find on our website, which I'm going to tell you about now. So as I mentioned, all of the curated data from our GCEPs is available on our website, clinicalgenome.org, and you can search for information multiple ways. You can use the search bar you see there. So you have gene selected, you can start typing in a gene, or you can go to what's highlighted in the orange box, gene disease validity, and you can select a list that you can search and filter by all the genes our GCEPs have curated. And that includes anything that's been recurated or reclassified. Next slide. So what I've done is pulled up just an example of what one of these gene disease validity reports would look like if you were to search the BRCA1 gene in the search bar on our website. So this includes the classification that was assigned, in this case definitive, the classification date, which is the date that the GCEP approved all of the evidence in the summary here, and as well as which of our gene curation expert panels completed this work. So I'll just show you at the bottom there. There's a lot of information on the screen, so you're not going to be able to see everything, but you'll see there's a few tabs at the bottom, and there would be the detailed evidence and how it was scored that the gene curation expert panel put together to come to the classification that they did. And I'll take this opportunity to note that the information provided on our website is not intended for diagnostic use or medical decision-making without review by a healthcare professional. If you do find this information useful and you choose to look up information on our website, we do encourage you to do that in partnership with a healthcare professional who is familiar with your health history. What we provide is general information. Anything related to your healthcare should be evaluated with a team who's familiar with you, your family, and your personal health history. Next slide. So what I'll do now is try to put this in context to say how might a gene curation result that ClinGen has provided inform a genetic test result in a clinical scenario. So I'll give you a brief example here of how this information may be useful. You see a sample, very simple genetic test report here. The ones in real life, as you probably know, have many more words on them. But in the case here, a 10-year-old child visits a genetics clinic with their family with a diagnosis of neurodevelopmental differences. The family comes with a genetic test report from exome sequencing performed in 2018. At that time, a variant of uncertain significance was reported in a gene considered to be a candidate gene. And as Caitlin introduced at the beginning of this session, a candidate gene is considered a candidate or a gene of uncertain significance when there is uncertainty in the gene's role in disease. So the family asked if there was any new information on these results, given that this test was reported on over 5 years ago. Next slide. And if the doctor or genetic counselor searches that candidate gene in particular on the ClinGen website, they'll see that we have evaluated this gene-disease relationship, and there's a full report available here. It's now classified by ClinGen as definitive in contributing to syndromic intellectual disability. The gene-disease relationship was evaluated in 2021, which was 3 years after the individual's genetic testing report. So this could certainly contain new information that the laboratory didn't have at the time of their reporting. This updated information could provide enough evidence for the healthcare team to investigate if this is not just a candidate, but a potential cause of the child's diagnosis. And they could also reach out to the reporting lab to see if the 2018 test results could be reevaluated in light of the new information. Next slide. So because we provide an example that shows how this might work out, to clarify a genetic test result, what you see here is that, yes, in fact, this information may be enough for the lab to reclassify the variant they initially called a variant of uncertain significance, now likely pathogenic, because now the gene has a definitive relationship with disease. And these updated results could provide new information that the healthcare provider and the family can use to navigate the future. So with that, I'll just move on to the next section, next slide. And now we'll talk about, we've talked about candidate genes, we've talked about how ClinGen evaluates gene-disease relationships. We talked about how you can find that information on the website, but now the title of the webinar that got you here, how to track this information over time, and how can our ClinGen resource help you do that? Right, so that's what we'll walk through. So everything you'll see on the next few slides, you can find on clinicalgenome.org. I'm going to use screenshots here, and we'll also put a few links in the chat afterwards with some tutorials that walk you through a similar thing, so you could use that on your own if you're interested after the webinar. So on our website, we have the ability, we give users, any user who has an email address and is interested in joining, to create a dashboard account, which gives you the ability to track genes that ClinGen has curated and receive notifications when information related to that gene is updated. If you click on the word dashboard in the top right-hand corner that's highlighted here, next slide, you'll be taken to a page describing the key features of the dashboard account. And you'll also see that blue box where you can either create an account and log in for the first time or log in if you're already registered and you're a returning visitor. Next slide. And so now let's imagine you've created an account. You are now logged in. You'll see your account name in the top right-hand corner rather than the word dashboard or login. And my ClinGen dashboard would be personalized to your account. So what you're seeing here is what I have under mine. And this is something you'd only be able to see if you were logged in, if you had created that account. When you were logged in, it will unlock these features that allow you to follow a gene. Following a gene adds them to the list that you'll see demonstrated in that slide. And what it will do is prompt an email notification if anything is updated on that gene as far as what ClinGen has curated. So the button highlighted here, follow new gene, you can use that to select genes you'd like to receive updates about. So yes, exactly. If you go to the next slide, let's pretend we hit that button, a search bar will come up. And as you type, gene names will appear based on the letters you've entered, and a suggested list will be offered. You can select the gene you are interested in and add it to your dashboard. If you're interested in adding 2 or 3 or 5, you can just go back and repeat that process until your list is complete. And next slide. So I'll point out, just in case you're coming here with a different scenario or report that you're trying to follow that isn't just adding a single gene. So when you are logged into that dashboard and you're given the option to follow genes, there are 2 other options. So I'm just going to go into those briefly. So if you would like to follow all the genes in a given stretch of a chromosome, something we may refer to as a genomic region, you may be interested in doing this if you are following all of the genes encompassed by a copy number variant that Caitlin mentioned at the beginning of the presentation. You can do this by using the follow by region option, and that will ask you for a chromosomal location. That may be something that's on your genetic test report. It may not be. It varies what information laboratories include on a report. So it may be something, if you're interested in doing that, to work with your genetic counselor or geneticist on. The next way to kind of bulk add a number of genes of interest is to do it by expert panel, either a gene curation expert panel or variant curation that we didn't get into today. But this would be getting notified not just when a particular gene is updated, but when a gene curation expert panel updates any of their curations. So if you're interested in not a particular gene associated with cardiovascular disease, but all genes associated with cardiovascular disease, you may select to get updates from the hereditary cardiovascular gene curation expert panel, for example. So next slide, Caitlin, thank you. And I'll show you one other feature. I'll note if you're logged into the website, you don't have to go directly to that dashboard to add genes to your follow list. If you also happen to be searching on the website and you pull up search results for a particular gene, in the example you see here, there's FBN1, there is that follow gene star that I've highlighted with that orange box here. And if you click that star, it will turn green and the gene will be added automatically to your dashboard view. So as you visit genes on the website, you can also follow or unfollow here using that star feature. Next slide. Thank you. And what I'm showing you here is an example of the notification that you may receive. So also in the dashboard, you can set your notification preferences, how often you'd like to receive notifications. And what you will get is this pretty straightforward email. So what you won't get, let's say now, is an email that tells you exactly what changed. What you will get is a notification that says there has been an update to a gene you are following, and the gene will be listed. It will prompt you to log into your dashboard account, and there you can find a summary of what changed. It won't highlight the specific information that changed, but the type of information. Was it a classification update? Did the disease name change, which can sometimes happen? And given that bit of information, you can go visit the curation report and look for the information that's been updated based on the type of change that was listed. Next slide. So those are the basics of how the dashboard will work. There's, like I said, some tutorials that will take you through that step by step and also talk about more of the functionality, what kind of information you can set your preferences on, things like that. So we'll share that with you now. And I just wanted to run through that with the screenshots. I also have it ready to go live if anyone has any particular questions that they have based on that discussion that you'd like to navigate through, we can do that. But I think that concludes the slides I put together.
Hi, thank you, Danielle. Thank you so much. We have one question about someone has a specific question about their dashboard, and it says that it's dominated by documents that they uploaded. Is that something that you have seen in ClinGen?
So without knowing much more, I do wonder if you were referring to the GenomeConnect dashboard versus the ClinGen dashboard. As far as I know, we don't have the ability to upload documents on the clinicalgenome.org dashboard. So it may just be a difference in these different websites. If indeed it's your clinicalgenome.org dashboard, I am happy to work with you further offline to get to the bottom of that.
That's a great possibility. So I just provided the info@genomeconnect.org email address. So if you have specific questions, please email us there, and then I'm happy to address them. So on a case-by-case basis and provide a little bit more clinical context or personal context based on that information. We did receive another question about mitochondrial genes and are there the same rules as with nuclear genes? So I'm going to answer the first bit of information. And so for the nuclear DNA, yes, there can be variants, so copy number variants or sequence variants. The main difference in the name is that the C dot is an M dot, and that M dot stands for mitochondrial. So that would be the big change that we would see in terms of the report that you would find. Is there are, Danielle, specific tracking for mitochondrial genes on ClinGen?
Yeah, so I will say there is the ability to follow any gene, regardless of if it is a nuclear gene or a mitochondrial gene. We do have a mitochondrial gene disease expert panel. So you can search by the under gene disease validity on our top search bar. You can search by expert panel, so you could review all of the information that they have provided there. Now, for mitochondrial genes specifically, they may have had to tweak or alter the framework for any differences, but that would be noted in the gene disease validity report and summary.
And I'm sorry, I was unable to see who asked the question about mitochondrial disease. So if there are additional questions that you had, please put them in the chat so we'll make sure that we can answer all of them. And the other thing that I will say, one of the things I forgot to mention for what GenomeConnect does do is we share individuals that maybe have a variant identified in a candidate gene with a resource called GeneMatcher. And GeneMatcher is a platform organization that tries to connect everyone around the globe that's studying certain candidate genes to try and put as many cases together in hopes of connecting researchers and clinicians to share information in order to help with that gene disease relationship classification as well. And so, as I mentioned, that participants enrolled in GenomeConnect that share their report and opt to receive updates if you do have a variant in a candidate gene and that gene has been associated with a disease, that is an update that we should provide back to our participants as well. So Jules just put in the chat, which was a slide that I jumped through real fast. And that's basically, if you have questions after today, you can email us at info@genomeconnect.org. If you have questions about your specific genetic test results, we do encourage you to reach out to the clinician that ordered the testing, or you can find a local genetics provider. And we put that link in the chat at the find a genetic counselor .nsgc.org. And so this is also the QR code to help find a local genetics provider. As Danielle mentioned, ClinGen is a resource that provides general guidelines and helps to provide rule books. It is not a source of medical information in terms of how to manage specifically your care and what treatments and screening that you should specifically have. That would be best discussed with your medical provider. As we said, to put all those pieces together, your personal health history, your family's health history, the change that was identified, all together to provide your best care. Okay, I just wanted to say a big thank you to Danielle. Thank you again for attending. Thank you for sharing your expertise. Thank you to all those that attended. Thank you to all that have participated and answering questions. And we have just put on one more link in the chat. And this is a survey for future topics that we should discuss in our webinars. So please, if you have ideas, if you like to hear something on a specific topic, I put some ideas in, but please put in your comments and the others. We're hoping to do more webinars in 2025. We really started these interactive webinars this year, and so we did 5 in 2024, and I think they hopefully have been a success, and we would love to hear from all of you as the people who are attending, what you want to know, what you want to learn, what other topics we may cover, or you have any other ideas in terms of format for interacting with all of you, please let us know. Thank you all so much and have a great day.